السلام عليكم. بسم الله الرحمن الرحيم. After cleavage of the zygote, the rate of cell division slows and normal cell cycle is restored. Morphogenesis. It means the process by which cells occupy their appropriate locations. It involves gastrulation. It means the movement of cells from the blastula surface to the interior of the embryo. And organogenesis, it means the formation of organs. Gastrulations rearranges the cells of the blastula into the three-layered embryo called the gastrula. So during the second week of embryogenesis, you remember that the embryo is named the bilaminar germ disc. It consists of two layers, epiblast and hypoblast. Whereas during gastrulation, there is a transformation of this bilaminar disc into trilaminar germ disc. It's composed of three layers, so it's named the gastrula. Now, this figure shows the blastocele. This is the layer of trophoblast, and this is the structure is named the blastula. During blastulation, where the two layers appear, the embryoblast and trophoblast. Here, the gastrula, so it changes from blastula to gastrula, where it's composed of three germ layers, the mesoderm, ectoderm, mesoderm, and endoderm. This figure shows the earlier stage of development, where there is the germ disc composed of two layers, epiblast and hypoblast, amnion cavity, yolk sac cavity, and this is the endometrium, where this embryo is embedded or implanted. Okay, this is the blastocyst cavity, and as I said, this is the bilaminar germ disc. It's composed of two layers. Now, the gastrulation, it begins with the formation of a structure named the primitive streak on the surface of the epiblast. This epiblast will be named the ectoderm later on. This is the primitive streak. It's clearly visible as a narrow groove with slightly bulging regions on either side at the caudal end of the embryonic disc. This is the embryonic disc. This area is the caudal end where we see the primitive streak. This arises at about 15 to 16 days. The cephalic end of the streak, the primitive node. It consists of a slightly elevated area surrounding a small primitive pit. So we have primitive node. This area is named the primitive pit, and this is the primitive groove. It consists of primitive groove, node, and pit, as you can see in this figure. After primitive streak formation, the embryo has craniocaudal axis, dorsal and ventral surfaces, right and left sides. By the end of the fourth week, the primitive streak shows regressive changes. It rapidly shrinks and later it disappears. Now this figure shows again, just to remind you, the stages of embryogenesis. This is the fertilized ovum or the zygote, starts cleavage. This is the 8-cell stage, and then it reaches morula. Then later on the blastula will be formed, or the blastocele, and then it will start. This invagination occurs, starting to enter the third week of development, and the gastrulation starts here. Here again, this is the blastocyst embedded inside or implanted in the endometrium of the uterus. This is the bilaminar germ disc, epiblast and hypoblast. As you can see in this figure, here the bilaminar germ disc appears clearly here. This figure, it shows the appearance of the third germ layer, the mesoderm. So we have ectoderm, which was previously named the epiblast, mesoderm, and endoderm. Endoderm, it was named the hypoblast. Here the gastrulation is completed, so there is change from bilaminar germ disc, two layers, into three layers, So the germ disk here is the trilaminar structure. The three layers produced by gastrulation are called embryonic germ layers. Ectoderm forms the outer layer, endoderm lines the digestive tract, whereas the mesoderm partly fills the space between the endoderm and ectoderm. Each germ layer contributes to specific structures in the adult animal. The most characteristic event occurring during the third week, as I named, is gastrulation. So we define the gastrulation process is the process that establishes all three germ layers in the embryo. The endoderm is the innermost, or this is the endoderm, and the middle layer is the mesoderm, the outer layer is the ectoderm. Now we are going to talk about the mechanism of gastrulation. During this process, cells of the epiblast migrate toward the primitive streak. Upon arrival in the region of the streak, what will happen? These cells become flask-shaped. They detach from the epiblast and slip beneath it. This inward movement is known as the invagination. Once the cells have invaginated, some will displace the hypoblast, creating the embryonic endoderm, and others come to lie between the epiblast and the newly created endoderm to form the mesoderm. Then the cells remaining in the epiblast then form the ectoderm. Thus, the epiblast, through the process of gastrulation, is the source of all of the germ layers, and cells in these layers will give rise to all of the tissues and organs in the embryo. This figure illustrates this mechanism here. This is the primitive streak, okay, and this is the dorsal surface or the superior surface of the germ disk. And here you can see this arrow point to the direction of migration of the cells, or these cells of the epiblast, which are close to the primitive streak. They detach from the epiblast, they enter this site and start migration, entering the area between the epiblast and hypoblast. Some of these cells will displace the hypoblast, forming the endoderm. This is the hypoblast. In this area, the hypoblast cells are displaced and replaced by the migrating cells forming the endoderm. In between them, some cells remain here. They are giving the mesoderm. The remaining cells of the epiblast will give the ectoderm. Here again, also the mechanism illustrated. This is the Hence's node of the primitive node. This is the primitive streak. And these arrow point again to the direction of migration of these cells, which are close to the primitive streak. As you can see here, the direction of migration, some of them displaced the hypoblast, forming the endoderm. These represent the mesoderm cell layer, which is named the intra-embryonic mesoderm, since we have extra-embryonic mesoderm. This is more clearly illustrating this process. Here again, this is the primitive streak, where the site of migration of cells is seen clearly. These cells displacing, this is the hypoblast, is displaced, forming the endoderm. This will form the intraembryonic mesoderm. Since we have extraembryonic mesoderm, these will meet together. So there will be fusion between intraembryonic mesoderm and extraembryonic mesoderm. And here, the remaining cells will form the ectoderm. As you remember, at the beginning, the blastocele is composed of two cell population, the trophoblast and the embryoblast. And this is then, during the second week, we have two cavities, amniotic cavity, yolk sac cavity. We have two layers, epiblast and hypoblast. We have trophoblast, which later on will divide into trophoblast and sensatio-trophoblast. Here again, this is another figure, shows the direction of migration of the cells from the epiblast to this area, which lies between the epiblast and the hypoblast. As more and more cells move between the epiblast and the hypoblast layers, they begin to spread laterally and cranially. Gradually, they migrate beyond the margin of the disc and establish contact with the extra-embryonic mesoderm, covering the yolk sac and amnion. In the cephalic direction, they pass on each side of the prochordal plate. This prochordal plate forms between the tip of the notochord and the oropharyngeal membrane. It's derived from some of first layers that migrated through the node in the midline and move in a cephalic direction. The oropharyngeal membrane at the cranial end of the disc consists of small region of tightly adherent ectoderm and endoderm cells that represents the future opening of the oral cavity. This figure shows again, this is the bilaminar germ disc, the primitive streak at the caudal end of the embryo. As you can see, these arrows represent the direction of the migration. You can see these are dotted arrows since the migration occurs between the epiblast and the hypoblast. Here, these cells will settle down. Some of them will form the intraembryonic mesoderm, some displace the hypoblast, forming endoderm, and some remain in the epiblast, forming the ectoderm. Now, each of these cell layers give derivatives, since all the structures of the embryo will arise from these layers. For example, the ectoderm layer or the outer layer gives epidermis of the skin and its derivatives as sweaty glands or hair follicles, nervous and sensory systems, pituitary gland, adrenal medulla, jaws and teeth, and germ cells. Whereas the middle layer, which is the mesoderm, gives skeletal and muscular systems, circulatory and lymphatic systems, excretory and reproductive systems, dermis of the skin, and adrenal cortex. The inner layer, the endoderm, gives rise to epithelial lining of the digestive tract and associated organs, for example, liver and pancreas. Epithelial lining of respiratory, excretory and reproductive tracts and ducts. Thus, thymus, thyroid, and parathyroid glands. Thank you for your listening.